topic for today, no, is DNA sequencing. So this is the second to the last topic for midterms, no. So since parehas lang naman yung topic natin for both LEC and lab, no, isahan na lang yung discussion natin sa laboratory. But syempre, pagdating sa quiz, no, sa quiz sa lab, ang focus natin is more of the procedure. And pagdating sa lecture, more on the theoretical part. Okay? So DNA sequencing is a sequencing method for determining the order, yung pagkakasunod-sunod ng mga nucleotide bases natin sa isang DNA molecule. So pag sinabi nating nucleotide bases, of course we have four, no? The basic building block of our DNA, it's adenine, guanine, cytosine, and thymine. Okay, so in the clinical laboratory, the DNA sequence information or yung pagkakasunod-sunod ng mga nucleotides sa DNA molecule is used routinely for a variety of purposes. So first, no, in detecting mutations, no. So kapag nagkaroon ng replacement or deletion, no. So for example, yung adenine, no, instead of sa guanine siya nakabind, no, nagbind siya sa cytosine. So that is one kind of mutation. So, pwedeng mag-cause yun, no, ng iba't ibang sakit. So, next, typing microorganisms. So, for example, no, nagkakaroon tayo ng mga different variants or serotypes for viruses. So, pwede nating i-detect yung mga DNA sequences nila para ma-differentiate yung old serotype from a new serotype. Okay, so more of infectious organisms to, no, yung mga uh, bacteria, viruses, parasite, and fungi. Okay. Next, identifying human haplotypes, no, and designating polymorphisms or more of mutations to. Wait. So next, we have targeted therapies which will be directed at abnormal DNA sequences detected by this technique. So more of procedure sa treatment naman to. Okay? So we have four methods. So first, manual sequencing. So under manual sequencing, we have Maxam-Gilbert method. Okay? So etong dalawang to, ito talaga yung mga pioneers pagdating sa DNA sequences. Okay? So silang dalawa yung magiging focus natin for today. Next, we have automated sequencing, which is actually a modernized method of Sanger. Okay, so ang principle niya is still based on Sanger, pero ginawa na siya sa automation. Okay, so meron na tayong software na ginagamit dito. Okay, next, you have pyrosequencing and bisulfite sequencing. Okay, so discuss natin yan isa-isa. So first, for Maxam-Gilbert method, it is developed by Alan Maxam and Walter Gilbert in the year 1970s. So it is also called as chemical sequencing method. Okay, so bakit siya tinawag na chemical sequencing method? Because it involves the use of different chemical agents or yung mga tinatawag nating base modifiers which are responsible for cleaving a specific nucleotide. So, bawat chemical agent, bawat base modifier, no, meron silang target na specific nucleotide na binibreak or kinikleave nila. Okay? So, it requires one double-stranded or single-stranded version of the DNA region to be sequenced with one end radioactively labeled. Okay? So, sa DNA sequencing, no, may radioactive label pa rin tayo. So kung maaalala ninyo yung radioactive label na ginamit natin sa hybridization-based technique, ano nga ulit yon? What is the radioactive label used for hybridization-based technique? Class, anyone? What is the radioactive label used for hybridization-based technique? Okay. It's an element, a radioactive element. Nakalimutan na. Class, wala pang 1 month ang nakalipas nung prelims ninyo. So, paano kung cover to cover yung finals? Aaralin nyo ulit siya mula sa prelims? Okay, it's the radioactive phosphorus or our 32P. Okay? So, siya din yung ginagamit na radioactive label for both Maxam-Gilbert and Sanger method. Okay? Tandaan, 32P, the radioactive label. Okay? Radioactive phosphorus. So, for sequencing, the labeled fragment or template is aliquoted into 4 tubes. Pag sinabi nating aliquoted, dinidivide natin yung sample natin, no? Yung liquid into 4 small volumes into 4 different tubes. Okay? And each aliquot is treated with a different chemical with or without high salt. So, eto na yung mga base modifiers natin. So, bawat tube, no, dun sa DNA sample natin na dinivide natin sa 4, lalagyan natin sila ng iba't ibang chemical agents or base modifiers which targets different nucleotides. Okay? Next, upon the addition of a strong reducing agent, so for the reducing agent, we use 10% piperidine and it facilitates the breaking of our nucleotides. Okay? So aside from our chemical agents or base modifiers, meron din tayong reducing agent. Okay? So this reducing agent, no, breaks the single-stranded DNA at a specific nucleotide. Okay? So tutulungan niya yung chemical agent or our base modifiers. Okay? So after the reactions, the piperidine is evaporated and the contents of each tube are dried and resuspended in formamide for gel loading. So the fragments are then separated by size on a denaturing polyacrylamide gel or yung tinatawag nating page. Okay? So ano ulit yung detection method natin na gumagamit ng electric current at gel para ma-separate yung mga fragments natin according to their size. Class. Okay, it's gel electrophoresis. Okay, so quiz nyo yan mamaya. Imposible hindi nyo alam yan. Okay, so we use gel electrophoresis to detect and to visualize, no, the fragments in Maxam-Gilbert method. Okay, so actually, same din kay Sanger. Gumagamit din tayo ng gel electrophoresis. Okay, at ang ginagamit nating gel is polyacrylamide. Okay, so the entire process lasts for 1 to 2 hours for short fragments and 7 to 8 hours for longer fragments. So for the components, we have the double-stranded DNA or the single-stranded DNA template, our reducing agent, the 10% piperidine, which is attached at the 5' end of our DNA. Next, we have the base modifiers, okay, or yung ating mga chemical agents. So, we have 4, okay, kasi di ba inalicode natin yung sample natin into 4, no? So, first, we have dimethyl sulfate. Second, we have formic acid. Third, we have hydrazine. Fourth, we have hydrazine with salt, okay? So, for the gel naman, no, the ideal concentration that we use is 6 to 20%. Okay? So, we use 6% polyacrylamide gel, pero pwede din siya hanggang 6 to 20%. Okay? So, next, we use loading dyes, no, para ma-detect natin sila at ma-visualize for the gel electrophoresis. So, pwede tayong gumamit ng bromophenol blue or xylene cyanol. Okay? Next, for our chemical agents or yung ating mga base modifiers, so discuss natin isa-isa. So first, we have dimethyl sulfate. Dimethyl sulfate, ang action niya is that it methylates guanine. Okay? So the reaction happens in 4 minutes at 25 degrees Celsius. Okay? So, ang target ni dimethyl sulfate, it's guanine. Okay? Next, we have formic acid. So, ang action, no, ni formic acid is that it protonates purine. Okay? At sino yung mga purine bases natin? Class, sino yung mga purine bases natin? Ano, nandyan pa ba kayo? Okay, we have adenine and guanine. Nasa PowerPoint na, no? So, purines, we have adenine and guanine. So, their action happens in 5 minutes at 25 degrees Celsius. So, ang target ni formic acid is both guanine and adenine. Next, we have hydrazine with salt. So, ang ginagawa naman niya is that it splits cytosine rings. Okay, so ang target ni hydrazine with salt is cytosine. So, the reaction happens at 8 minutes, no, at 25 degrees Celsius. Next, we have hydrazine. No? So, ang ginagawa niya is that it splits pyrimidine rings. At sino yung mga pyrimidine natin? Klase, open niyo yung mga mic ninyo. Okay, so we have cytosine and thymine. Okay? So still, the reaction happens at 8 minutes, no, at 25 degrees Celsius. Okay? So, paano ba yun, ma'am? So, first tube natin, di ba sabi natin, meron tayong dimethyl sulfate. So, sabi natin, it methylates guanine. So, the chain, no, our sample, the template, breaks at guanine. Okay? So, mapuputol yung DNA strand natin at guanine. Okay? So, etong kulay yellow na to, this is your label. Okay? The 32P. Okay? The radioactive label. Okay? So, for dimethyl sulfate, ang na-target niya is guanine. Okay? Next, we have formic acid, which protonates purines. Okay? So that the chain breaks at guanine and adenine. Okay? So, mapuputol yung sample natin at adenine and guanine. Okay? Next, we have hydrazine, which targets our pyrimidine rings. Okay? So, thymine, cytosine. Okay? The chain will break at thymine and cytosine. Okay? Next, we have hydrazine with salt. Tina-target naman niya is cytosine rings. Okay? So, the chain will break at cytosine. Okay? So, anong magiging itsura nun? So, first, aliquot muna natin yung sample natin into 4 tubes. So, DNA sample sa first tube, DNA sample sa second tube, DNA sample sa third tube, DNA sample sa fourth tube. Okay? And for tube 1, ang ilalagay natin is dimethyl sulfate. Tube 2, formic acid. Tube 3, hydrazine. Tube 4, hydrazine with salt. Okay? So, yung DNA sample natin, aliquoted into 4 tubes. And yung 4 tubes na yun, lalagyan natin ng base modifiers which targets a different nucleotide. Okay? So, the denaturing conditions prevent the single strands of DNA from hydrogen bonding. So, may nilalagay pa tayo na formamide, urea, and heat, no? Para ma-prevent natin na mag-bind ulit sa isa't isa yung mga single strands of DNA natin. Okay? So, para hindi na sila mag-form ng hydrogen bond ulit. Okay? Para kapag i-gel electrophoresis natin sila, they will migrate strictly according to their size, no? Doon sa size ng mga nucleotides nila. Okay? So, the sequence is inferred from the bands on the film. Okay, so in interpretation natin, di ba, for the gel electrophoresis is the presence of the bands on our film. So the smallest band, okay, so the smallest, it's the fastest migrating, represents the band closest to the labeled end of the fragment. Okay, yung labeled end natin, it's the 5 prime. Okay, di ba yung 32P natin nakabind sa 5 prime. Okay, the lane in which the band appears identifies the nucleotide. So kung ano yung band present, yun yung nucleotide present. Okay, so the sequence is read from the bottom at the 5 prime end. Okay, magsa-start tayo sa baba, papunta sa taas sa 3 prime end. Okay? So for the interpretation, pag may band present for guanine and adenine and guanine, no, it is read as guanine. Okay? If there is a band present only in guanine and adenine, it is read as adenine. Okay? If there are bands present for both cytosine and thymine and cytosine, it is read as cytosine. And if the band is only present in cytosine and thymine, it is read as thymine. Okay? So yung tubes natin kanina with different base modifiers, ilalagay natin yan sa gel electrophoresis. So sa tube 1, no, ilalagay natin siya sa well 1. Okay? Tube 2 sa well 2, tube 3 sa well 3, tube 3 sa well 4. Okay? And each tube, di ba, meron silang mga target na base, no, or mga nucleotides. So in this case, sa well 1, it's the guanine. Next, sa well 2, it's guanine and adenine. Well 3, cytosine and thymine. Well 4, cytosine. Okay? So interpretation tayo. Example, for example, meron tayong band present in guanine and adenine. Okay? So may band present in guanine and adenine. Ano yung nucleotide present natin?
Adenine po.
Okay, it's adenine. Okay? Next, for the two bands, we have a band present for both cytosine and thymine and cytosine. Okay? So ano yung nucleotide present natin?
Cytosine.
Okay, cytosine. Next, we have bands present for guanine and adenine. Ano yung band or the nucleotide present?
Guanine po.
Okay, it's guanine. Last, we have cytosine and thymine. So, ano yung nucleotide present?
Thymine po.
Okay. So, hanggang sa mapunta tayo sa 3 prime. Okay, so laging magsa-start sa baba, sa 5 prime, papunta sa taas, no, sa 3 prime. Okay? So, and so on, hanggang sa mapunta tayo sa 3 prime. Okay, nakuha po yung interpretation.
Yes po.
Yes po, ma'am.
Okay, so isang example pa. So first, meron tayong band present for cytosine and thymine. Ano yung nucleotide present natin?
Cytosine po.
Cytosine po.
Cytosine. So pwede din natin siyang isulat horizontally. So 5' dash cytosine. Okay? Next, we have bands present for cytosine and thymine.
Thymine po.
Thymine. Next, adenine and guanine.
Guanine.
Adenine.
Adenine po.
It's adenine. Next, cytosine and thymine.
Guanine po, ma'am.
Ha?
Cytosine po.
Cytosine po.
Ano naging guanine? Cytosine. Okay? Next, we have bands present for adenine and guanine.
Guanine po.
Guanine po.
Okay, guanine. Next, we have bands present for cytosine and thymine.
Thymine
Thymine
po.
Thymine. And lastly, we have adenine and guanine.
Adenine.
Adenine po.
We have adenine dash 3 prime N. Okay? So, check natin yung answer natin. We have cytosine, thymine, adenine, cytosine, guanine, thymine, adenine. Okay. So, nakuha, class? So, next week, ganito yung magiging quiz ninyo, ha? Maglalabas ako ng picture, tapos, isusulat nyo yung order ng mga nucleotides. Okay?
Paulit nga po, ma'am.
So, dun tayo sa interpretation. So, kapag merong bands present for guanine and guanine, it is read as guanine. Okay? Kapag meron tayong band present for guanine and adenine, it's adenine. Band present for cytosine and thymine and cytosine, it's cytosine. Okay? Band present for cytosine and thymine, we have thymine. Okay? Nakuha, Christian?
Yes po, ma'am. Thank you po.
Okay. So, summarize lang natin yung procedure natin. So, first, the double-stranded DNA is denatured into a single-stranded DNA. Next, we prepare for tubes, okay? And pipette our single-stranded DNA sample to each test tube. So, i-aliquot natin siya into 4 tubes, okay? Next, we add the required chemical on each test tube. So, sa first tube, we have dimethyl sulfate. Second tube, we have formic acid. Third tube, we have hydrazine. And fourth tube, we have hydrazine with salt. Okay, so okay lang naman magbali-baliktad sila, no? Basta alam nyo kung anong target ng bawat chemical agent or base modifiers natin. Okay? Next, we add the reducing agent at 90 degrees Celsius, no, to facilitate the breaking of the strand. So ano ulit yung ginamit nating reducing agent? We have 10% ano? 10 Okay, 10% piperidine. Okay, so that is our reducing agent. Okay? Next, we proceed to polyacrylamide gel electrophoresis or our page to separate the fragments based on their size. So sa well 1, ilalagay natin yung sample natin sa tube 1, well 2, tube 2, well 3, tube 3, well 4, tube 4. Okay? Then we proceed to autoradiograph, no, para ma-detect and to visualize, no, yung mga fragments natin. So, ita-transfer natin yung mga bands from the gel to an X-ray film. Then, we read the sequence starting from the bottom at the 5' end, going to the top sa 3' end. Okay? So, limitations for Maxam-Gilbert method. So, although Maxam-Gilbert method or sequencing is a relatively efficient way to determine short runs of sequence data, the method is not practical for high-throughput sequencing of long fragments. So, for shorter sequences, okay pa siyang gamitin, but for longer fragments, no, medyo matrabaho na siya. So, it is not practical to use Maxam-Gilbert method for long fragments. Okay? So in addition to that, we use hazardous chemicals such as hydrazine and piperidine, our reducing agent, which requires more elaborate precautions for use and storage. So Maxam-Gilbert method has been replaced by the dideoxy chain termination sequencing method or the Sanger method. Okay, so Sanger method replaced Maxam-Gilbert method. Okay, so discuss natin si Sanger method. Before that, wala na bang questions kay Maxam-Gilbert method? Nakuha ba? Class, any questions?
Wala po.
Okay. Next, kay Sanger method. So it is discovered by Frederick Sanger in the year 1977. So it is also known as the chain termination method or the dideoxy chain termination sequencing method. So bakit siya tinawag na dideoxy tsaka chain termination, no? Because we use dideoxynucleotides, no? Our DDNTPs which stops, okay, so stops termination, the DNA synthesis at specific nucleotide. So it is ideally used for DNA base pairs of less than 1,000. So mas malaki na yung range natin pagdating sa mga sequences ngayon. Okay? And it gives 99.9% base accuracy and considered as the gold standard for DNA sequences. Okay, so it is a modification of the DNA replication process. Okay, so just like our PCR, ginagaya niya yung DNA replication process in vivo or inside our body. So we still use a short synthetic single-stranded DNA fragment or our primers, which are of course complementary to our target sequences. Okay? So for the components, we use a single-stranded DNA template, M13 bacteriophage as our primer, our DNTPs, no, our deoxynucleotides used for the synthesis, DDNTPs for termination, and our fluorescent dye or our radioactive dye, no, the 32P or the radioactive phosphorus. Okay? So ano bang kinaibahan no, ng ating DNTPs from our DDNTPs? Okay, so kung titingnan nyo no, sa kanyang carbon atom, at the third carbon atom, we have hydroxyl group for DNTPs. Okay? Pagdating kay DDNTPs, wala na tayong oxygen, walang hydroxyl group. Okay? So this hydroxyl group is required in the formation of a phosphodiester band with another nucleotide. Okay? So kapag wala na siyang hydroxyl group, ibig sabihin hindi na tayo makakapag-bond, no? Hindi na makakagawa ng phosphodiester band sa isa pang nucleotide. Okay? So kapag ang DDNTP nag-bind sa area natin to be sequenced, no? Magsi-stop na dun yung synthesis. Wala nang pwedeng mag-bind na iba. Kasi wala nang formation ng phosphodiester band. Okay? Kasi wala nang oxygen eh. Walang hydroxyl group. Okay? So, ang mangyayari dyan, kung ito yung ating template, this is the area to be sequenced. We have our primer. So, syempre, mag initiate siya ng synthesis. We have the DNA polymerase. No? And then, magbabind yung mga DNTPs natin. Okay? But for example, meron tayong DDNTP. In this case, ang DDNTP natin is DDATP. Okay? Just like our DNTPs kasi class, no? Our DDNTPs, meron din tayong 4 kinds, no? Our DDATP for adenine, DDCTP for cytosine, DDGTP for guanine, and DDTTP for thymine. Okay? So for example, yung DDATP natin nag-bind dito kay guanine, di ba? Kasi sila yung complementary, adenine and guanine. So if nag-bind si DDATP kay guanine, no? And si DDATP wala siyang hydroxyl group. Ibig sabihin kapag nag-bind si DDATP dun sa area natin na sinisequence, wala nang ibang pwedeng mag-bind pagkatapos niyang kumabit kay guanine. Okay? Kasi wala siyang hydroxyl group. Hindi na siya makakapag-form ng phosphodiester band sa isa pang nucleotide. Okay? So wala nang makakapag-bind dito onwards. Okay? So kay DDATP, magsa-stop na yung synthesis. Okay? So each DDNTPs target a specific nucleotide. Okay? So for the procedure, first we have the double-stranded DNA is denatured into 2 single-stranded DNA. So we have a mixture of a template and primer, 1:1, okay? And is placed into 4 separate reaction tubes in sequencing buffer. So i-divide din natin yun into 4 tubes. So the sequencing buffer is usually provided with the sequencing enzyme and contains ingredients necessary for the polymerase activity. Okay, so kailangan natin yung DNA polymerase, no, para ma-start yung ating reaction. Next, the mixture of all four DNTPs are added. Okay, so pagdating sa DNTPs natin, yung 4 ilalagay natin, no, yung ating DATP, DCTP, DGTP, and DTTP. Okay? So DNTPs natin, yung 4 ilalagay natin sa bawat tube. Okay, dun sa 4 na tube. Pagdating sa DDNTPs natin, ang ilalagay lang natin is one of the 4. Okay? And we have a different DDNTP in each of the 4 tubes. Okay? So, kung sa first tube, DDATP lang ilalagay natin. So, in this tube, ang target natin is guanine. Okay? Next, ang ilalagay natin is DDCTP. Target natin is cytosine. Okay? Next, DDGTP, ang target natin is guanine. And DDTTP, ang target natin is thymine. Okay? So, with the addition of DNA polymerase enzyme, no, in the 4 tubes, the reaction begins. Okay? So, we use the DNA polymerase, no, to start the reaction. And after about 20 minutes, no, the reactions are terminated by the addition of a stop buffer. Okay? So, the stop buffer terminates the reaction. Okay? So, anong laman ng stop buffer natin? It is consists of 20 millimolar EDTA, which chelates cations and stop enzyme activity. Next, we have formamide, which denatures the product of the synthesis reaction. And of course, our gel loading dice, no, para ma-visualize natin yung ating mga fragments pagdating sa gel electrophoresis. Okay? So we have bromophenol blue and xylene cyanol. So any of the two pwedeng gamitin. Okay? So ganito yung magiging itsura or setup ng mga tubes natin. So first we have the sample. So wala kasi yung sample. Yung sample natin. Next, we have the primer. Okay? DNA polymerase, which will start the reaction. Next, we have our DNTPs. Okay? So, lahat yung 4 na DNTPs present sa bawat tube. Okay? At pagdating sa ating mga DDNTPs, no, ang meron lang is isa sa bawat tube. Okay? At iba-iba sila per tube. So, DDATP sa first tube, so on, hanggang 4th tube. Okay? So the sets of synthesized fragments are then loaded onto a denaturing polyacrylamide gel. So polyacrylamide pa rin yung ginagamit natin pagdating sa electrophoresis. Okay, yung page natin. So the products of each of the sequencing reactions are loaded into 4 adjacent lanes. Okay, so 4 wells sa ating gel, okay, and they are labeled as A, C, G, or T corresponding to the DDNTP in the 4 reaction tube. Okay, so A for our DDATP, G for our DDGTP, and so on hanggang sa thymine natin. Okay? So once the gel is dried and exposed to an X-ray film or yung ating autoradiograph, the fragment patterns can be visualized from the signal on the 32P labeled primer. So the 4-lane gel electrophoresis pattern of the products of the 4 sequencing reactions is called the sequencing ladder. So the ladder is read to deduce or to interpret the DNA sequence or yung order ng mga nucleotides natin. So it is still read from the bottom going to the top. So yung nasa pinakababa, it is the smallest and the fastest migrating fragment. Okay, so ito yung closest dun sa primer natin or dun sa ating label. Okay, so start tayo sa 5' end sa baba, going to the top sa 3' end. Okay, so unlike our Maxam-Gilbert method na yung mga base modifiers natin, yung iba tinatarget nila dalawang nucleotide, no? Kasi sa purines and pyrimidines yung reaction nila. Pagdating kay Sanger method, since we use different DDNTPs, no, very specific na yung presence ng mga bands natin. So kung may band present kay cytosine, no, ang nucleotide present natin is cytosine. Kung may band present tayo for adenine, ang nucleotide present natin is adenine. May band present for guanine, we have guanine. Band present for thymine, it's thymine. Okay? So, mas direct na siya. Okay? Very specific na kasi yung mga DDNTPs natin. Okay? So, example. So, in this case, meron tayong band present for adenine. state yung mga nucleotides present natin. So, start tayo sa 5' end. Okay? So, 5', meron tayong band present for adenine. Ano yung nucleotide present?
Adenine.
Okay, we have adenine. Next band.
Guanine.
Okay,
Guanine.
guanine. Next,
Cytosine.
we have cytosine. Next,
Tapos,
dito na tayo.
Guanine.
Kunin po.
Okay,
guanine.
guanine. Next,
Thymine po.
thymine. For the next 3,
Cytosine po.
Cytosine po.
okay, 3 cytosine. Next, ito muna.
Thymine po.
Okay, thymine. Then next,
Adenine.
2 adenine. Next,
Guanine
Guanine.
po.
guanine,
Thymine po.
Thymine po.
thymine,
Cytosine.
Cytosine po.
cytosine,
Adenine po.
Adenine po.
2 adenine,
Cytosine.
Cytosine po.
cytosine.
Guanine po.
Next,
Thymine po.
thymine. And last,
Guanine po.
Kunin po.
okay, guanine dash 3'. If we check natin, we have adenine, guanine, cytosine, guanine, thymine, 3 cytosine, thymine, 2 adenine, guanine, thymine, cytosine, 2 adenine, cytosine, thymine, guanine. Okay. So, nakuha po yung Sanger method.
Yes po, ma'am.
Okay. So, any questions so far?
Wala po.
Okay.
Nag-attendance na po ba, ma'am?
Hindi pa. Okay, so method, no? Yun yung na-discuss natin kanina. So, bago magamit tayo ng 4 test tube with a DDNTP each. Okay? So, next, for modern method, no? Sa modern method, gumagamit tayo ng fluorescent label instead of a radioactive label, okay, to each DDNTPs. And they are added to a single tube alone. So, kung kanina sa classic method, meron tayong 4, no, na wells. Okay? So, yung 4 na well para sa isang sample lang. Okay? Ngayon, kay modern method, ang ginawa nila, each DDNTP has a corresponding color. Okay? Fluorescent dyes yung ginagamit. Okay? So, pwede siyang ilagay sa isang diretsong well lang. Okay? So, isang well, isang sample. Unlike kanina kay classic method, mag-occupy tayo ng 4 na wells, okay, para sa isang sample. So, for modern method, ang tinitignan is yung color nila. Okay? So, each DDNTP has a designated color. Okay? So, yun yung modernized method. Yun yung automated fluorescent sequencing natin. So, this is the modern method of Sanger sequencing. Okay? So, it uses double-stranded DNA templates and cycle sequencing. So, machine na yung gumagana dito. Okay? So because cycle sequencing does not require sequential additions of reagents to start and stop the reaction, the cycle sequencing is more easily adaptable to high throughput applications and automation. So unlike kanina sa manual sequencing natin, 'di ba nag-add tayo ng DNA polymerase to start the reaction. Then after 20 minutes, maglalagay tayo ng stop buffer. Okay? So for automated fluorescent sequencing, no, hindi na kailangan mag-add after 20 minutes, no? Lahat 'yon yung machine na yung gagawa, no? Even sa monitor, okay? Andun na yung DNA sequence natin. Hindi na natin kailangan tingnan yung gel electrophoresis. Titingnan natin isa-isa kung ano yung bands present. Ano yun, Christian?
Ma'am, paano po pag colorblind yung tumitingin po sana na yan?
Hindi siya pwedeng mag-interpret. Tsaka isa pa, hindi mo na kailangan tingnan yung kulay kasi yung machine na yung maglalabas. No? Hindi na tayo mag interpret. Even the interpretation is automated. Okay? So, hindi siya human na titignan kung anong kulay. Okay? Yung machine na yung titignan doon. Or yung titingin doon. Okay?
Thank you po, ma'am.
Okay, so we use fluorescent dyes instead of our radioactive nucleotides, no, to label the primers or sequencing fragments. So fluorescent dyes used for sequencing have distinct colors or peak wavelengths. Okay, so ang tinitingnan na natin dito is wavelengths, no? Pero pwede din naman yung bands, no? But some machines or automations, mas marami yung gumagamit ng wavelengths. Okay, so the advantage of having 4 distinct colors is that all 4 reaction mixes can be read in the same lane of a gel or on a capillary. Okay, so paano yung sa capillary? So instead of using a gel, no, for wells, gumagamit na lang tayo ng capillary electrophoresis, which is also one kind of electrophoresis. No? And mabivisualize natin siya sa kulay nila. So in this case, galing kasi to sa book, no? So yung kulay niya is more of gray, black, and blue lang, no? Pero yung kulay talaga nila is more of blue, yellow, red, and ano pa ba? Basta later, sasabihin ko lang. Okay, so in this case, adenine is gray, guanine is light blue, thymine is black, cytosine is dark blue. Okay, so ang tinitignan na lang natin dito is the color. Okay? Para ma-identify natin yung sequence nila. Okay? So we have two methods of sequencing. First is the dye primer. Okay? So sa dye primer, this is the same in the manual method, no? Wherein our fluorescent dye or yung label is attached to the primer at the 5' end. Okay, kaya siya tinawag na dye primer kasi yung dye naka-attach sa primer. Okay? Next, we have dye terminator. Okay? So, naka-attach naman yung fluorescent dye natin sa DDNTPs natin. Okay? Kaya siya tinawag na terminator. Kasi 'di ba yung terminator natin dito is the DDNTP. Okay? So naka-attach naman siya at the 3' end. Okay? So this is an example of a dye terminator. No? So the label fragment synthesized according to terminal end of the sample. So this is the is color green, cytosine is color blue, DDGTP, red as guanine, is black or yellow, DDTTP, red as thymine, is color red. Okay? So, ang babasahin natin, yung peak wavelength. Okay? Yung pinakamataas na wavelength. Okay? So, in this case, dito sa baba, makikita nyo, meron ding mga wavelengths dyan, pero ang babasahin natin is yung peak, yung pinakamataas. Okay? So, for black, no, we have DDGTP, so it is guanine. Green, okay, adenine. Red for thymine, then blue for cytosine. Okay? Ganon siyang ini-interpret. Okay? Pero ayan, makikita nyo yung machine na yung maglalabas ng sequence. No? So, hindi problema kung colorblind ka. Okay? So, for the sequencing interpretation, both dye primer and dye terminator sequencing are loaded onto a slab or capillary gel. Okay, so we use an electropherogram, no, to interpret. So it shows the sequence signal frequency, which requires a software para ma-interpret niya kung ano yung peak na wavelength present. Okay, so the quality of electropherogram depends on the quality of the template, efficiency of the sequencing reaction, and the cleanliness of the ladder. Okay? So, na-gets class yung automated sequencing. So, any questions for automated fluorescence? Wala? Okay, so proceed tayo with pyrosequencing. So pahapyawin lang natin to, no? So pyrosequencing, it is the most useful for short to moderate sequence analysis and there is no need for a sequencing ladder. So it relies on luminescence, no? Light when nucleotides are added to a growing strand of DNA. So madalas siya na ginagamit for the detection of mutation and single nucleotide polymorphism. So ginagamit din siya for HLA typing. So pag sinabi nating HLA, this is the human leukocyte antigen. So it is tested, no, to detect compatibility between the donor and the recipient during organ transplant. Okay, so that is HLA typing. So pwede din tayong gumamit ng sequencing methods for that test. Okay? So for the components of our pyrosequencing, first we have the single-stranded DNA template, our sequencing primer, sulfurylase and luciferase enzymes, our substrate which is adenosine 5' phosphate and luciferin. Okay? So pagdating sa pyrosequencing, wala tayong ginagamit na gel at wala ding fluorescent dyes sa DDNTPs. Okay? So, ang ginagamit talaga natin pagdating sa detection is the presence of or the generation of light or luminescence. Okay? So, once a DNTP forms a phosphodiester band with a primer, it releases pyrophosphate, okay, that will be converted to ATP. So, very familiar kayo dyan. ATP, yung ginagamit natin for energy by the help of sulfurylase in the presence of our pyrophosphate substrate. So, ATP is used to generate luminescent signal by the enzyme luciferase. Okay? So, it is a luciferase catalyzed conversion of luciferin to oxyluciferin. So the reaction of each DDNTP is added one at a time in the flow cell and observed for light peaks. Okay, so wavelength pa din tayo dito. So the sequences are interpreted through a pyrogram. So, paano ba nangyayari yon? Ito yung diagram natin. So, first, we have our DNA and DNTP. So, kapag nagkaroon ng phosphodiester band sa dalawa, using a DNA polymerase enzyme, no, makakapag-produce tayo ng pyrophosphate. Okay? Pyrophosphate, kapag naman inad natin yung substrate natin, yung adenyl sulfate, no, using the enzyme sulfurylase, makakapag-produce tayo ng ATP. And ATP, using the enzyme luciferase, will convert luciferin to oxyluciferin and will produce light. Okay, kapag is the same enzyme in the far field, nag-act yung enzyme luciferase, makakapag-produce tayo ng light. Kasi meron tayong energy in the form of ATP. Okay? So, ang dinedetect natin is the formation or the generation of light. Okay? So, the DNTPs are added one at a time using a flow cell. Okay? At tinitignan natin yung light peaks. Okay? So, kung merong na-produce na peak, ibig sabihin present yung nucleotide na pinasok natin sa flow cell. Okay? Lastly, we have the bisulfite DNA sequencing. So it is the detection of DNA methylation, which is a modification of the chain termination method or our Sanger method. Okay? So ang dinetect natin dito is more of the epigenetics, wherein walang alteration sa DNA sequence, pero merong change in the phenotype. Okay? So pag sinabi nating methylation, it is the addition of methyl group to cytosine which inactivates, no, the gene. So in the presence of bisulfite, the methylated cytosine will remain as it is even if nag-add tayo ng bisulfite, no? While the unmethylated cytosine will be replaced by uracil and eventually change to thymine. So cytosine remains as it is, will be counted as methylated, and those that are changed to thymine will not be counted. So for bisulfite DNA sequencing, ang tinitingnan natin dito is kung ilan, no, yung mga methylated cytosine natin. Okay? Ilan yung methylated cytosine? So, for example, in this case, meron tayong black rat. Okay, meron tayong black rat meron tayong white rat. Okay? So they have a different phenotype. Pag sinabing phenotype, this is the physical characteristic of one organism. Okay? So in this case, yung phenotype nila na yon, it's the color. Okay? So black yung isa, white yung isa. Pero parehas sila ng DNA sequence. Okay? So parehas na parehas yung mga base pairs nila. Okay? Pero yung isa white, yung isa black. Okay? So anong nagkakos nun, ma'am? Okay? We have methylated cytosine. Okay? May mga nag-bind na methyl groups dun sa cytosine nung white rat, okay, activates again. Kaya nagkaroon siya ng change in the physical characteristic. Okay? So from black, naging white siya because nagkaroon ng methylation. Okay? So in genetics, no, para magkaroon tayo ng change in phenotype, kailangan muna natin ng alteration in the DNA sequence. So pwedeng magkaroon ng deletion, okay? Or magkaroon ng replacement. For example, yung adenine nag-bind kay guanine. Dapat complementary, di ba? But for example, si adenine nag-bind kay cytosine, no? That will cause an alteration in a DNA sequence. Okay? So that is mutation. Okay? And that will cause a change in the phenotype of the organism. Okay? But in epigenetics, no, there is no alteration in the DNA sequence, but we have a change in the phenotype. Okay? So that is caused by the addition of a methyl group in the cytosine. Okay? Which inactivates a gene. Kaya nagkakaroon tayo ng change in the phenotype. Okay? So for bisulfite sequencing, ang dinedetect natin is the methylated cytosine. Okay? So in the addition of bisulfite, okay, the methylated cytosine will remain as methylated cytosine even if meron na tayong inad na bisulfite. Okay? While the unmethylated cytosine will be changed to uracil and eventually change to thymine. Okay, so cytosine mag-change to uracil then to thymine in the presence of bisulfite. Okay? While the methylated cytosine, may mga naka-attach na methyl group, kahit mag-add tayo ng bisulfite, cytosine pa rin sila. Hindi sila magiging thymine. Okay? So, ang binibilang natin dito, it's the methylated cytosine. Okay? So, in this case, we have two. No? So, methylated cytosine. Even if nag-add na tayo ng bisulfite, still cytosine. Okay? While in this case, yung mga unmethylated cytosine, they are converted to uracil, then eventually to thymine. Okay? So, ang bibilangin natin, yung mga naging cytosine pa rin, even if nag-add tayo ng bisulfite. Okay? So, yun yung bilang ng mga methylated cytosine. Okay? So, nakuha class yung bisulfite DNA sequencing. Class?
Yes po.
So, any questions? Okay. So, for our references, no, ang for this page tuloy. Chapter 10, I think, ng Buckingham. Then, for Shen, more of additional notes lang tayo. Okay, so that ends our second to the last topic for Mod X for both Lec and Lab. So, tomorrow, sa lecture time, no, wala na tayong discussion. So, diretsong quiz na tayo nung week 8, other amplification techniques. Okay? So, next week, meron pa rin tayong quiz for both Lec and Lab, no? So, sa laboratory, more of procedures yung focus natin. Then, for the lecture, yung theoretical part. Okay? So, meron na din akong pinublish na unit task for DNA sequences sa laboratory. Okay? So, pakisagutan na lang din yun. So, for your quiz, no, yung week 7 and 8 ng ating laboratory, no, naka-publish na din siya. So, pwede nyo na lang i-take yung quiz off-cam para makapag F2F classes na kayo. Okay? So, no need to enter our Gmeet. Pakisagutan na lang yung quiz. Okay? Pero i-check ko yung mga Canvas activity ninyo. So, once na meron tayong nakitang red mark, so automatically, hindi ko siya i-record. Okay? So, wala nang questions. Wala nang questions.
Wala na po.
Okay. So, check ko lang anong oras.